Hello and welcome. If you are settling in for the evening, finding your position, letting the quiet gather around you, letting the events of the day begin to soften, then you have come to exactly the right place. This is the Science Channel for Sleep. And tonight, we are going somewhere that very few human beings have ever been. We are going down, past the sunlit surface of the ocean, past the last traces of blue twilight, into a world of permanent darkness, extraordinary pressure, and surprising, patient, extraordinary life. Tonight, we are exploring the strange creatures of the deep ocean. Before we begin, if you enjoy this kind of calm, unhurried journey through the natural world, if this is the sort of thing you like to fall asleep to or rest quietly alongside on a still evening, it would mean a great deal if you took a moment to like this video and subscribe to the channel. It truly helps more than you might expect, and it helps this channel continue making exactly this kind of content. And if you feel like leaving a comment, we would love to know where in the world you are listening from tonight and what time it is there. It is always interesting discovering where listeners are tuning in from around the world. Now, let us go down. The deep ocean, defined as any water below 200 meters, where sunlight can no longer fuel photosynthesis, is the largest living space on Earth. It contains more than 95% of all the habitable volume available to living things on this planet. It is divided into zones. The mesopelagic twilight zone from 200 to 1,000 meters. The bathypelagic midnight zone from 1,000 to 4,000 meters. The abyssopelagic zone from 4,000 to 6,000 meters. And the Hadal zone of the deep trenches below 6,000 meters. Each zone is darker, colder, and more pressurized than the last. And each zone, it turns out, is inhabited. The creatures that live in these zones face three fundamental challenges that have shaped everything about them. Darkness, complete, permanent, absolute. Pressure, increasing by 1 atmosphere for every 10 meters of depth, reaching over 1,000 atmospheres in the deepest trenches. And food scarcity, because no plants grow in the deep ocean without sunlight, and most deep-sea creatures depend on the slow rain of organic material drifting down from the world above. These three pressures have produced organisms unlike anything in the sunlit world. Organisms of extraordinary strangeness and extraordinary elegance. Not monsters, simply solutions. Biological solutions to biological problems, refined by hundreds of millions of years of evolution in one of Earth's most stable and challenging environments. Interestingly, approximately 76% of deep-sea organisms produce bioluminescence, their own biological light. The deep ocean is not completely dark. It is lit by living things, by flashes and glows and pulses of blue and green and occasionally red light produced by the organisms themselves, for purposes of hunting and defense and communication that we are only beginning to understand. Right now, as you rest, the deep Pacific Ocean is alive with these displays. The deep Atlantic, the deep Indian Ocean. In the permanent darkness of the midnight zone, creatures are producing light, hunting, drifting, growing, going about the slow, patient business of deep-sea life, with no human observer present, as they have been doing for millions of years. New deep-sea species are discovered at a rate of approximately 2,000 per year. most of the deep ocean remains unexplored. The creatures we will visit tonight are only the ones science has so far been able to reach and study. Let us begin. For most of human history, the deep ocean was not merely unexplored. It was imagined to be empty. In 1843, a British naturalist named Edward Forbes proposed what became known as the azoic hypothesis, the idea that below approximately 550 meters, the ocean was completely lifeless. Forbes had conducted careful dredging surveys in the Aegean Sea and had noticed that organisms became progressively scarcer as depth increased. He drew a straight line through his data and extrapolated to zero, concluding that at some point, life simply stopped. The hypothesis made intuitive sense to many scientists of the era. The deep ocean was dark, cold, and subject to crushing pressure. What could possibly live there? The first cracks in the azoic hypothesis came not from scientific expeditions, but from engineering. In the 1860s, when the early transatlantic telegraph cables were retrieved from the ocean floor for repair, they were found to be encrusted with organisms, sea life growing on cables that had been sitting at depths Forbes had declared lifeless. Something was living down there, and it was using human infrastructure as a substrate. The definitive answer came from one of the most ambitious scientific voyages in history. The HMS Challenger left Portsmouth in December of 1872 on a 4-year voyage around the world's oceans. Equipped with dredges, trawls, water sampling equipment, and thermometers, the Challenger systematically sampled the ocean at depths throughout the Atlantic, Pacific, Indian, and Southern Oceans. What came up in the dredge nets demolished the azoic hypothesis permanently. Life was everywhere, at every depth sampled, in forms no one had imagined. The Challenger expedition collected thousands of new species, organisms from the deepest accessible parts of the ocean, and the scientific reports published over the following decades formed the foundation of modern oceanography. Deep-sea biology had begun, but knowing that the deep contained life was very different from being able to see it. The specimens brought up in nets were dead, compressed, often deformed by the pressure change as they were hauled to the surface. They were shadows of the living creatures they had been. That changed in 1930, when an engineer named Otis Barton and a naturalist named William Beebe descended together into the Atlantic Ocean off the coast of Bermuda in a steel sphere approximately 1.5 meters in diameter. They called it the bathysphere. It was lowered on a cable from a surface ship and could not move independently. If the cable broke, they would be lost. It had 3 small portholes of fused quartz, and through these portholes, Beebe looked into the deep ocean and described what he saw via telephone cable to a radio audience at the surface. What he saw was extraordinary. Bioluminescent creatures drifting past the porthole in the darkness, forms he had never seen. In 1932, Beebe and Barton reached a depth of 923 meters, a world record at the time. Beebe described many specific creatures in detail through his porthole, some of which were later confirmed by science, and some of which were never confirmed, and may have been products of an excited imagination peering through a small, dark window into an alien world. The research submersible Alvin which began regular dives in the 1960s, transformed deep-sea biology. Capable of independent movement, equipped with cameras and sample collection tools, Alvin allowed scientists to observe creatures in their natural environment, alive, behaving normally, undisturbed by the violence of a net haul. It has completed more than 5,000 dives. In 1977, its passengers discovered something that changed biology. But that discovery belongs to a later section. The giant squid, Architeuthis dux, illustrates how long it can take to truly know a deep-sea creature. Known for centuries as the Kraken, a sea monster large enough in legend to sink ships, the giant squid was real enough that fragments of tentacles and dead individuals washed up on beaches. But it was not photographed alive in its natural deep-sea habitat until 2004. A Japanese research team, working with a baited camera line, lowered to approximately 900 meters depth in the Pacific, captured the first photographs of a living giant squid. In 2012, a different Japanese team, using specialized low-light cameras, captured the first live video footage. For decades before that, science knew the giant squid primarily from the beaks found in the stomachs of sperm whales, a creature defined for a long time by what ate it. The colossal squid, Mesonicoteuthis hamiltoni, heavier than the giant squid and found in Antarctic waters, was not known to science at all until 1925, when tentacles with rotating hooks were found in a sperm whale's stomach. A complete specimen has never been filmed alive. Remotely operated vehicles, ROVs, have brought a new era to deep-sea biology since the 1980s. Unlike submersibles carrying human passengers, ROVs can stay at depth for extended periods, enter spaces too small or dangerous for crewed vehicles, and observe creatures without the disturbance of a human presence. It was ROV footage that finally revealed the true nature of the barrel-eye fish's transparent head, misunderstood for decades from dead specimens. And it is ROV footage that continues season after season to show scientists creatures behaving in ways that dead specimens could never have revealed. Researchers believe the current rate of deep-sea species discovery, approximately 2,000 new species described per year, will continue for generations. The deep ocean is not a closed book. It is a book most of whose pages have not yet been opened. And what those pages contain, in terms of creature diversity and biological strangeness, is something worth exploring slowly and carefully, one extraordinary adaptation at a time. The first and most pervasive of those adaptations is one that transforms the very nature of the deep ocean environment. It turns out that the deep ocean is not dark. That is where we are going next. The deep ocean is dark. This is true in one sense. No sunlight penetrates below approximately 200 meters in any meaningful quantity, and below 1,000 meters, no sunlight reaches at all. But to say the deep ocean is dark in the way that a closed room is dark, completely, uniformly, without any light at all, would be to miss one of the most remarkable features of life at depth. Approximately 76% of deep-sea organisms produce bioluminescence. 3/4 of all the creatures in the deep ocean carry their own light. The deep sea is not dark so much as it is lit by a different kind of light. Biological light, cold light. light produced not by any star or lamp, but by the chemistry of living things. Bioluminescence is produced when a compound called luciferin is oxidized in the presence of an enzyme called luciferase. The reaction releases energy as light with almost no heat, a cold blue or green glow produced precisely and efficiently by organisms that have refined the chemistry over millions of years of evolution. It is, in its basic mechanism, not so different from the chemical light sticks sold at camping stores. The principle is the same. The biological execution is vastly more sophisticated. Most deep-sea bioluminescence is blue or green. These wavelengths travel furthest through seawater. They are absorbed least by the water itself, allowing them to carry the furthest before fading. An organism producing blue bioluminescent light can be detected at a greater distance than one producing red or yellow light. And in the deep sea, where distances are important and encounters are rare, every meter of detection range matters. But blue and green are not the only colors in the deep sea's palette. A small number of deep sea organisms produce red bioluminescence. And this is where the story becomes particularly interesting. Most deep sea organisms have eyes adapted to detect blue and green light, the wavelengths that dominate their environment. Red light, being rapidly absorbed by seawater, does not occur naturally in the deep ocean, and the eyes of most deep-sea creatures have not evolved the photoreceptors to detect it. Red, in the deep sea, is effectively invisible. This means that a creature capable of producing red bioluminescent light possesses what is effectively a private flashlight, a light it can use to illuminate its environment and its prey while remaining completely undetected by anything around it. The black dragonfish and the stoplight loosejaw are among the creatures that have evolved this remarkable capability. The black dragonfish produces red bioluminescence from organs below its eyes and may also have red-sensitive photoreceptors, allowing it to see what it illuminates. The stoplight loosejaw produces both red and green bioluminescence, two private wavelengths, an illumination system of considerable sophistication in an organism that is barely 20 centimeters long. The uses of bioluminescence extend well beyond the private flashlight. Counter-illumination is one of the most elegant applications. Many fish in the mesopelagic zone, the twilight zone between 200 and 1,000 meters, have rows of light-producing organs called photophores along their undersides. These photophores produce a soft, downward-directed glow that precisely matches the faint residual sunlight filtering down from above. From below, a fish using counter-illumination has no silhouette. Its bioluminescent underside blends seamlessly with the background light, rendering it effectively invisible to predators looking upward. Defense takes other forms as well. The Atolla jellyfish, a disc-shaped deep-sea jellyfish, produces a spinning circular display of blue bioluminescent light when it is grabbed or disturbed. This display functions as what scientists call a burglar alarm, a brilliant, sudden illumination that attracts larger predators to investigate the disturbance. A predator grabbing an atolla may find itself attacked by a larger predator drawn to the light show, and the atolla, released in the confusion, escapes. The bioluminescent display is not a weapon, but a distress signal aimed at drawing danger toward the danger. A deep sea worm called swimmer bombiviridis, sometimes called the green bomber, takes this strategy in a different direction. When disturbed, it detaches small posterior segments from its body, tiny glowing green capsules that drift away from the worm in the water. The pursuing predator turns toward the glowing decoys while the worm retreats into the darkness. The segments are sacrificed, but the worm survives, and the segments will regenerate. The firefly squid of Japan, Watasinia scintillans, produces spectacular bioluminescent displays across its entire body and comes to shallow water in Toyama Bay each spring to spawn in such numbers that the displays are visible from boats at the surface. In the deep sea, where it spends most of its life, these displays serve communication and possibly mate attraction. Interestingly, bioluminescence has evolved independently many times across many different lineages of deep-sea organisms. Fish and squid and jellyfish and worms and bacteria and single-celled organisms have all developed the ability to produce light through separate evolutionary paths. The chemistry is slightly different in different lineages. The light-producing organs have different structures, but the result is the same. Light produced by a living thing in the darkness of the deep ocean. Researchers believe this convergent evolution reflects how important and how useful bioluminescence is in the deep sea environment. Useful enough that evolution has found its way to the same solution many times in many different organisms. The deep ocean, then, is not dark. It is a world of living light, of flashes and glows and pulses that constitute their own ecology, their own communication system, their own way of navigating the permanent night. Organisms are hunting with light, hiding with light, signaling with light, and defending themselves with light. Understanding that the deep ocean is lit by its own inhabitants changes the picture of what it is like to live there. Because the next question is, beyond the darkness and the light, what does it feel like physically to be a deep-sea creature? The answer involves pressure, cold, and some of the most elegant biological engineering on Earth. That is where we are going next. To understand what it means to be a deep-sea creature, it helps to begin with the numbers. Pressure in the ocean increases by 1 atmosphere for every 10 meters of depth. At the surface, the pressure is 1 atmosphere, the weight of the air column above us, which we carry without noticing because our bodies evolved within it. At 100 meters, the pressure is 11 atmospheres. At 1,000 meters, the beginning of the midnight zone, it is 101 atmospheres. At the bottom of the Mariana Trench, the deepest point in the ocean at approximately 10,935 meters, the pressure reaches approximately 1,086 atmospheres. At those pressures, water itself is measurably compressed. The water at the bottom of the Mariana Trench is approximately 5% denser than water at the surface. The pressure is so great that it affects even the most incompressible of ordinary substances. For living things, the challenge is not the pressure itself, but what pressure does to biological molecules. Proteins, the workhorses of all biological chemistry, responsible for every enzymatic reaction, every structural element of cells, every signal that passes between one cell and another, are sensitive to pressure. High pressure causes proteins to fold incorrectly, to lose their shape, and to lose their function. A surface organism placed at deep sea pressure would find its proteins deforming, its membranes stiffening, its cellular machinery ceasing to function. It would not simply be crushed, it would fail biochemically. Deep-sea organisms have evolved an elegant solution. The molecule is called trimethylamine oxide, TMAO. It is a small organic molecule that counteracts the pressure-induced dysfunction of proteins by stabilizing their three-dimensional shapes under high pressure. It works as a kind of molecular pressure suit, a chemical chaperone that keeps proteins properly folded even when the physical environment around them is trying to force them out of shape. Researchers have found that the concentration of TMAO in deep-sea fish tissue increases proportionally with the depth at which the fish normally lives. A fish from 200 meters has a certain TMAO concentration. A fish from 2,000 meters has a higher concentration. A hadal fish from 8,000 meters has a very high concentration indeed. The molecule can be used almost like a depth gauge. Measure the TMAO in a fish's muscles and you have a good estimate of where it normally lives. The gas-filled swim bladder, the organ that allows most surface fish to control their buoyancy by adjusting the volume of gas inside, presents a different pressure problem. Gas is compressible, and under deep-sea pressures, a gas-filled bladder would be crushed to a tiny fraction of its surface volume, rendering it useless for buoyancy control. Many deep-sea fish have solved this problem simply by abandoning the swim bladder entirely. They are negatively buoyant, denser than water, and sink when they stop swimming. Others have replaced the gas in their swim bladders with oil, which is not compressible, providing neutral buoyancy without the pressure vulnerability of a gas-filled bladder. The cold of the deep ocean, typically 1 to 4 degrees Celsius, presents its own challenges. Cell membranes are made of lipids, and at cold temperatures, lipids become less fluid, more rigid. The membrane can stiffen to the point of malfunction. Deep-sea organisms produce antifreeze proteins and cold-adapted lipid compositions that maintain membrane fluidity at near-freezing temperatures, keeping their cellular machinery running in conditions that would freeze or stiffen the membranes of surface organisms. Gelatinous bodies, the jelly-like substance that makes up the bodies of jellyfish, ctenophores, and many deep-sea invertebrates, are nearly incompressible. Water transmits pressure without structural damage, and a body composed primarily of water is far better suited to high-pressure environments than one containing air-filled spaces or rigid structures that could crack or compress. One reason so many deep-sea organisms are gelatinous is that this body plan is inherently pressure-resistant. Coloration in the deep sea follows its own logic. Many deep-sea organisms are black, absorbing any ambient light without reflecting it, becoming as invisible as possible in the darkness. Others are red. Red pigmentation at depth appears black because red wavelengths of light do not exist in the natural deep-sea environment. They are absorbed by seawater before reaching these depths. A red animal in the deep sea is, to the eyes of most deep-sea predators, an invisible one. Scientists have found that the Mariana snailfish, the deepest living fish known, observed at over 8,000 meters, is nearly white, almost entirely without pigmentation. In the absolute darkness of the Hadal zone, color is irrelevant. There is no light to reflect or absorb, no predator with eyes adapted to any wavelength. The energy that would otherwise go into producing pigment is available for other uses. Some deep-sea fish have evolved extraordinary eyes, very large, with specialized retinas adapted to detect the faintest traces of bioluminescent light. Others have lost their eyes entirely, relying instead on other senses: vibration detection, chemical sensing, the lateral line system that detects water movement. In an environment where vision is entirely useless, every feature of a deep-sea creature's body is a response to the conditions of its environment. The gelatinous form, the chemical composition of its tissues, the presence or absence of a swim bladder, the color of its skin, the size and structure of its eyes, all of it shaped by the same pressures: darkness, cold, and the weight of kilometers of water above. And all of it shaped also by a fourth pressure that we have not yet addressed directly, the pressure that may, in some ways, have been the most creatively generative of all: the pressure of hunger. That is where we are going next. In the sunlit surface ocean, food is relatively abundant. Phytoplankton bloom in the warmth and light. Zooplankton graze on the phytoplankton. Small fish eat the zooplankton, and so on up the food chain. A productive, interconnected community, sustained by the energy of the sun. In the deep ocean, there is no sun, no phytoplankton bloom, no photosynthesis of any kind. The deep sea depends, for almost all of its food, on what drifts down from above. This is marine snow, the slow, gentle, continuous rain of organic material from the surface ocean to the seafloor. Dead organisms, fecal pellets, shed mucus, fragments of plankton and zooplankton, all of it aggregating into particles and drifting downward through the water column, taking weeks to months to fall from the surface to the abyssal plain. By the time it arrives, most of the energy it originally contained has been consumed by organisms in the mesopelagic zone. What reaches the deepest seafloor is a fraction of what fell from the surface. For deep-sea creatures, food encounters are rare, unpredictable, and precious. Every calorie matters. And this fundamental fact of deep-sea life has driven some of the most extraordinary evolutionary adaptations on Earth. The first and most pervasive response is the slowing of metabolism. In the cold, dark deep, with no need to generate heat and no need to be constantly alert in a world where encounters of any kind are infrequent, deep-sea organisms have reduced their metabolic rates to the minimum needed to sustain life. They move slowly, they grow slowly, they live long. A slow metabolism means that a single meal can sustain an organism for weeks or months, stretching the value of every food encounter to its maximum. The giant isopod, Bathynomus giganteus, a deep-sea relative of the common pillbug that can reach 50 centimeters in length, has been documented surviving in captivity for more than 5 years without eating. Its metabolism is so slow and its energy reserves so carefully managed that it can endure extraordinary periods of food deprivation and emerge apparently unaffected to eat again when food eventually arrives. The orange ruffy, a deep-sea fish commercially fished in the southern oceans, can live for 150 years, partly as a consequence of its extremely slow deep-sea metabolism. Its slow life has made it extraordinarily vulnerable to commercial fishing. When populations are depleted, recovery takes not years but decades. But patience alone is not sufficient when food does arrive. When an opportunity presents itself in the food-scarce deep ocean, the organism that can capture the largest possible meal has the greatest advantage. And this has driven the evolution of some of the most remarkable prey capture structures in the animal kingdom. The gulper eel, Eurypharynx peleconoides, has a mouth so large relative to its body that it seems almost comically disproportionate. The lower jaw can hinge open so widely that the mouth becomes essentially a vast bag, capable of engulfing prey many times the size of the eel's body. The stomach is correspondingly expandable. The gulper eel can swallow very large prey and then slow its metabolism while digesting the enormous meal over an extended period. The black swallower, Chiasmodon niger, takes this approach even further. It can swallow prey more than twice its own body length and 10 times its own mass. Its stomach expands around prey far larger than the fish itself, the skin stretching over the swallowed animal like a transparent sack. Dead black swallowers have been found floating at the ocean's surface with their stomachs burst open, the prey inside too large to digest before decomposition set in, the expanding gases eventually rupturing the stomach wall. It is, in the most literal sense, an organism that sometimes dies from eating too ambitiously. But in a food-scarce environment, the evolutionary pressure toward maximum consumption is so strong that even this risk is apparently worthwhile. Large eyes, or the absence of eyes entirely, represent another food-related adaptation. Eyes are energetically expensive to maintain. In the deep sea, where food is scarce, the energy invested in maintaining large, complex eyes is justified only if those eyes provide a significant advantage. For many mesopelagic organisms, very large eyes allow detection of the faintest bioluminescent flashes from prey or predators, providing an early warning system or hunting advantage that justifies the metabolic cost. For organisms in deeper zones where there is no light at all, not even bioluminescence, eyes provide no benefit, and some deep-sea species have reduced their eyes to vestigial structures or lost them entirely, redirecting the energy savings to other purposes. Researchers believe that the deep sea's food scarcity has also driven the reduction of bone and muscle in many deep-sea fish. Bone is metabolically expensive to produce and maintain. It requires significant energy inputs. Muscle likewise. Many deep-sea fish have reduced skeletal mineralization and greatly reduced muscle mass compared to surface fish of similar size. Their bodies, stripped of everything non-essential, pared down to the minimum needed to function in a world where energy is always precious. The result is a body type, common in the deep sea, that appears almost insubstantial, soft, barely solid, almost ghost-like. Not because the deep has failed to provide material for solid bodies, but because solid bodies are an unnecessary luxury in an environment where every joule of energy must be accounted for. Food scarcity, then, has shaped the deep-sea body as completely as pressure and darkness. The slow metabolism, the enormous mouth, the expandable stomach, The minimal bone and muscle, the large eyes or absent eyes, all of it written in the biology of organisms that have been solving the problem of food scarcity for millions of years. No creature illustrates the intersection of these pressures more perfectly and more strangely than one that has become perhaps the most famous of all deep-sea animals. That creature is where we are going next. The anglerfish has become, in the popular imagination, a symbol of the deep sea, a creature so strikingly strange that its image appears in films and illustrations whenever the deep ocean needs to be represented. The enormous gaping mouth, the needle-like teeth, the glowing lure dangling from the forehead like a fishing rod grown from the fish's own body. This reputation is understandable. The anglerfish is genuinely extraordinary, but the popular image is also somewhat misleading in size, in character, and in the most astonishing aspect of anglerfish biology, which is not the lure at all, but the reproductive strategy that accompanies it. Let us begin with what an anglerfish actually is. Most anglerfish species, the deep-sea ceratioid anglerfish that carry the bioluminescent lure, are small. A common misconception imagines them as large, formidable predators. In fact, most species are between a few centimeters and about 20 centimeters in length for the females, about the size of a tennis ball to a fist. They are not large creatures. They are small, soft-bodied, and in the cold, dark water of the bathypelagic zone, they drift with minimal movement, conserving energy. The lure, the esker, is the structure that defines them. It is a modified dorsal fin spine, a fin ray that, over evolutionary time, has migrated to the top of the head and been transformed into a slender rod tipped with a small bulb. Inside that bulb live bioluminescent bacteria. The anglerfish provides the bacteria with nutrients. The bacteria produce the light. It is a symbiotic arrangement. Two organisms, bacterium and fish, each providing what the other needs. The esker glows blue in the darkness, a single point of light in the absolute blackness of the midnight zone, the only illumination in all directions for perhaps hundreds of meters in every direction. Small organisms, drawn to the light by curiosity or by instinct, approach. The anglerfish waits, and when the prey is close enough, the enormous mouth snaps open, the teeth designed not to cut, but to trap, curving inward so that anything that enters cannot easily escape. Some species can modulate the light of their escape, switching it on and off, creating a flashing signal. Whether this flashing pattern carries information, whether it is a language of sorts, is not known. It may simply make the light more attention-catching. In a world where almost any light is rare and significant, the distinction may not matter much. Now, the male. The female anglerfish, the fish described above, is the large one. The male of the same species is a very different creature. He is tiny. In many species, only a centimeter or two long. He hatches from an egg, develops his eyes and his nostrils at an extraordinary rate, and his entire biological purpose from the moment of hatching is to find a female and reproduce. The female is far larger than the male. Depending on the species, she can be 20 to 60 times his length. He is, relative to her, like a small toy placed beside the animal it represents. Finding a female in the vast dark ocean, at densities of perhaps 1 anglerfish per cubic kilometer of water or less, is the central challenge of the male's existence. Researchers believe he does this primarily by following chemical gradients, pheromones released by the female that diffuse through the water and allow him to navigate toward her source. The specific mechanism is not fully understood, but somehow, in the absolute darkness of the midnight zone, across distances that may be substantial, tiny males find females. When he finds her, he bites her. This is not a hostile act. It is the beginning of a union that will last the rest of his life. When the male bites the female's skin, enzymes are released at the contact point, enzymes that begin to digest the skin of both male and female simultaneously. The tissues fuse, blood vessels connect. The male's circulatory system merges with the female's. He is now receiving all of his nutrition through her blood. He will never feed independently again. After fusion, his eyes begin to degenerate. They are no longer needed, and maintaining them costs energy that the female's body now controls. His fins degenerate. His internal organs reduce. He becomes, over time, a pair of testes attached to the female's body by a connection of shared tissue and shared blood, providing sperm when she is ready to spawn. A single female may carry multiple males simultaneously, small nodules of tissue attached to her body, each one once an independent organism with its own eyes and fins and behavioral repertoire, now reduced to a single reproductive function. The initial scientific reaction to this discovery was one of considerable confusion. When small worm-like creatures were found attached to female anglerfish in the early 20th century, they were initially described as external parasites or juvenile fish of a completely different species. The idea that they were male anglerfish, permanently fused to the female, sharing her bloodstream, completely dependent on her for their survival, took time to establish and was, when established, one of the more remarkable findings in the history of deep-sea biology. The anglerfish's reproductive strategy is an extreme solution to an extreme problem, the problem of finding a mate in a vast, dark, sparsely inhabited ocean where encounters of any kind are rare. Evolution, given enough time and a strong enough selective pressure, produces solutions that would seem impossible if we had not seen them. The esker glows in the darkness. The female drifts. Somewhere in the water around her, a tiny male follows the chemical trail she has laid. It has been happening in the midnight zone for tens of millions of years, and the anglerfish is not the only deep-sea creature to have evolved solutions to survival that seem, at first, almost beyond belief. The next creature we visit is one that has been the subject of human mythology for centuries, finally photographed alive only 2 decades ago. That is where we are going next. In Norse mythology, it was called the Kraken. Sailors described it as an island-sized creature that rose from the sea without warning, a mass of tentacles large enough to drag a ship beneath the surface, a monster at the edge of the known world, beyond which the charts of medieval cartographers bore the warning, "Here be dragons." The Kraken was not entirely imaginary. There was something in the deep ocean that was, by any ordinary standard, enormous. Something that occasionally left evidence of its existence on the surface world, in the form of tentacle fragments on beaches, circular sucker scars on the skin of sperm whales, and the enormous beaked heads found in the stomachs of those same whales. Something very large, very real, and very difficult to observe. It would take until 2004 before a human being photographed it alive in its natural environment. The giant squid, Architeuthis dux, can reach lengths of up to 13 meters for the largest females, though most individuals are considerably smaller. It is the second largest invertebrate on Earth after the colossal squid, and it has the largest eyes of any living animal, eyes that can reach 30 centimeters in diameter. 30 centimeters, the span of an average human face from chin to hairline. Why eyes so large? The answer, researchers believe, lies in what those eyes are designed to detect, not the reflection of sunlight, which does not exist in the mesopelagic zone where the giant squid primarily lives, but bioluminescence. Specifically, the bioluminescent flashes produced by the skin of sperm whales as they move through the water. Sperm whales are the primary predator of the giant squid. They dive to mesopelagic depths, sometimes well below 1,000 meters, in pursuit of squid, which make up the majority of the sperm whale's diet. As a sperm whale moves through bioluminescent organisms, it disturbs them, producing flashes and trails of light that might be detectable at considerable distances to an eye sufficiently large and sensitive. The giant squid's enormous eye, on this interpretation, is an early warning system, evolved not to see the world in detail, but to detect the approach of a whale in time to attempt an escape. Every centimeter of additional eye diameter increases the light-gathering area and extends the detection range. 30 centimeters of eye diameter is the result of an evolutionary arms race between a squid that needs to detect approaching whales and a whale that needs to approach without being detected. The giant squid lives primarily at depths of 300 to 1,000 meters, the mesopelagic and upper bathypelagic zones. It was known to science from fragments and dead specimens for well over a century before it was ever photographed alive. In 2004, a Japanese research team, Tsunemi Kubodera and Kyoichi Mori, lowered a baited camera line to approximately 900 meters in the North Pacific. The photographs the camera returned were the first ever taken of a living giant squid in its natural habitat. The squid on the line was approximately 8 meters long. It fought the bait with considerable vigor before eventually breaking free, leaving behind a fragment of tentacle. In 2012, a larger Japanese expedition, using specialized low-light cameras developed specifically for the challenge of filming in the near-complete darkness of the deep mesopelagic, captured the first video footage of a living giant squid in its natural environment. The footage showed the animal at depths of 600 to 900 meters, alone, moving through the dark water. For all of recorded human history before that moment, no human being had seen a living giant squid behaving as a giant squid normally behaves in its own environment. 2,000 years of mythology, 150 years of scientific description, and the creature remained essentially unknown as a living animal until the digital age. The colossal squid, Mesonicotodis hamiltoni, is a different, less known, and in some ways even more remarkable creature. Found in the waters of the Southern Ocean around Antarctica, the colossal squid is heavier than the giant squid. the heaviest invertebrate on Earth, though not necessarily longer. Rather than suckers on its tentacles, the colossal squid has rotating hooks, swiveling barbed hooks that can grip and cut prey with considerable effectiveness, making it a more formidable predator than its more famous relative. The colossal squid's eyes may reach 27 centimeters in diameter, with built-in photophores, light-sensing organs embedded in the eye itself, that may allow the animal to detect its own bioluminescent output for orientation. It was known to science from fragments found in sperm whale stomachs beginning in 1925. A complete specimen was not recovered until decades later. The colossal squid has never been observed alive in its natural habitat. The mating behavior of both the giant and colossal squid has never been directly observed in the wild. The full reproductive cycle of neither species is known. The giant squid is encountered so rarely, even now, that fundamental questions about its ecology remain unanswered. It turns out that even the creatures most famous from the deep sea are still, in important ways, unknown. Their skin, at least, we understand somewhat better. Cephalopods, the group that includes squid and octopuses, have skin containing chromatophores, which are pigment-filled cells that can expand or contract to change the color of the skin, and papilla, which are muscular projections that can alter the texture of the skin surface from smooth to rough. These changes happen rapidly, within fractions of a second, and in deep-sea cephalopods, they may constitute a form of communication, conveying information about state or intention that researchers are only beginning to decode. The Humboldt squid, which visits deep water during the day and shallower water at night, has been observed producing bioluminescent flashes across its body surface that appear to be coordinated and information-bearing. Whether this constitutes a true language in any meaningful sense is debated, but it is a reminder that the deep sea's cephalopods are not simply large and strange. They are, by the standards of invertebrate life, remarkably sophisticated. The predatory strategies of the squid are formidable, but other deep-sea creatures have developed their own approaches to hunting in the dark. Creatures with structures so extreme that they make the anglerfish's lure seem almost understated. Those creatures are where we are going next. The food-scarce deep ocean has a particular way of shaping predators. When meals are rare, when a creature might go days or weeks or longer between feeding opportunities, the evolutionary pressure on every aspect of prey capture becomes intense. The mouth must be large enough. The teeth must be effective enough. The attack must be fast enough. And in an environment of permanent darkness, the sensory systems used to detect prey must be exquisitely sensitive. The result, across dozens of independent evolutionary lineages in the deep sea, has been a menagerie of predatory fish of extraordinary variety, each one a different solution to the same fundamental problem of capturing food in the dark. Let us visit a few of them. The viperfish, Charliodus sloani, is a slender fish of the mesopelagic and upper bathypelagic zones, typically between 20 and 30 centimeters long. What makes it immediately recognizable is its teeth. The viperfish's fangs are so long that they cannot be contained within the closed mouth. They curve upward and outward, extending beyond the upper jaw like curved transparent needles, visible even when the mouth is shut. When the viperfish opens its mouth, These fangs angle outward, creating a cage-like structure. A prey animal swimming into this space encounters the fangs as the mouth closes, impaled by teeth moving toward it from multiple directions simultaneously. But the viperfish does not rely only on passive encounter. It also has a bioluminescent organ on its elongated dorsal fin, a photophore that it holds extended forward over its own body like a fishing rod. The effect is to combine the active luring strategy of the anglerfish with the fang trap of its own mouth, two predatory strategies operating simultaneously in one small fish. The black dragonfish, various species of the genus Idiocanthus, takes a different approach to the darkness. Its body is entirely black, making it as visually invisible as it is possible to be in the deep ocean. But below each eye, it has a bioluminescent organ that produces not blue or green light, but red. As discussed in the section on bioluminescence, red light is invisible to most deep-sea organisms. The black dragonfish can therefore illuminate its environment, scanning the water with red light, without alerting anything around it to its presence. It carries a private lamp in a darkness where everyone else is blind to that color. Some dragonfish also have red-sensitive photoreceptors in their eyes, allowing them to see what they illuminate, completing the system. Scientists have found that the stoplight loosejaw, Malacosteus niger, goes even further. It produces both red and green bioluminescence, two private wavelengths, each invisible to most other deep-sea organisms. It essentially has two different private flashlights operating on two different channels. The sophistication of this system, in an organism that is less than 30 centimeters long and lives in complete darkness at depths of 500 to 1,000 meters, is remarkable. Many dragonfish species have a barbel, a whisker-like appendage that hangs from the chin, tipped with complex bioluminescent structures. These structures can include multiple light-producing organs of different colors and can be quite elaborate in their design. The function is luring. The barbel is waved in the water to attract prey toward the mouth. Some dragonfish have bioluminescent organs inside their mouths as well, potentially illuminating prey that has already been caught and allowing the fish to see what it is eating. The deep-sea dragonfish produces light inside its own mouth, one of the more intimate and specific uses of bioluminescence in the deep-sea toolkit. Then there is the fangtooth fish, Anoplagaster cornuta, whose name is, if anything, an understatement. The fangtooth has the largest teeth relative to body size of any fish in the ocean. Its teeth are so large that special accommodations have had to evolve for the fish to be able to close its mouth at all. The long lower fangs fit into corresponding sockets in the roof of the mouth, the upper fangs fitting into corresponding cavities in the lower jaw. The fish cannot fully close its mouth in the way that surface fish do. There is always a gap, the teeth interlocking in their accommodating cavities rather than the lips meeting. Interestingly, the fangtooth is not a large fish. Most individuals are between 10 and 16 centimeters in length. Its extraordinary teeth are packed into a relatively small body. The entire organism organized, it seems, around the apparatus for capturing prey. Its coloration is uniformly dark, a near black, that makes it effectively invisible in the deep ocean darkness. Some deep-sea shrimp have developed their own version of the escape response. When threatened, they can eject a cloud of bioluminescent fluid from glands near their mouths. The glowing cloud expands in the water around the predator, while the shrimp retreats into the darkness. A bioluminescent smokescreen that works not by hiding the shrimp, but by filling the water with distracting light. The predatory fish of the deep ocean are not, taken individually, the largest or most powerful predators that evolution has ever produced, but they are among the most precisely specialized organisms in which every structural feature reflects the specific challenges of hunting in cold, dark, food-scarce water, refined over millions of years to the point where little is wasted and everything serves a purpose. The deep sea's tendency to produce organisms of extreme specialization extends beyond the predators. Some of the strangest fish in the ocean are strange not because of their hunting apparatus, but because of the unusual shapes their bodies have taken in response to the pressures of the deep. Those unusual shapes are where we are going next. The deep ocean has produced fish of a variety of body forms that, taken collectively, suggest evolution working through a catalogue of possibilities without the constraints that apply in more competitive, better-lit environments. When you are adapting to darkness and pressure and food scarcity, the usual rules of fish design, streamlined body, forward-facing eyes, proportionate mouth, are less binding. Different solutions emerge. Some are solutions to predation. Some are solutions to vision. Some are solutions to finding mates in a sparse ocean. And some are solutions so unusual that they took decades of scientific observation to understand. The barrel-eye fish, Macropinna microstoma, is one of the most visually striking fish in the deep sea and one of the most misunderstood. For many years, the barrel-eye was known from dead specimens brought up in nets. Those specimens showed a fish with a somewhat ordinary-looking face, two eyes visible in roughly the position you might expect. What was unusual was the transparent dome covering the top of the head, filled with some kind of fluid. Its function was not understood. In 2009, researchers from the Monterey Bay Aquarium Research Institute, MBARI, observed living barrel-eye fish from a remotely operated vehicle and finally understood what had been misread for decades. The two round structures visible in preserved specimens were not eyes at all. They were olfactory organs, the equivalent of nostrils. The actual eyes were inside the transparent, fluid-filled dome, two large, green, tubular structures that were, in the living fish, facing upward through the dome. And crucially, the eyes could rotate. Barrel-eye fish spend much of their time with their eyes directed upward through the dome, looking for the silhouettes of prey against the faint downwelling light from above. When a prey item is detected and the fish moves to pursue it, the eyes rotate forward, pointing directly ahead to track the prey. The transparent dome protects the eyes, which are large and sensitive, from the stinging cells of the siphonophores, whose feeding tentacles the barrel-eye often moves beneath, stealing food items from the tentacles. Researchers believe the barrel-eye's green pigment in its eyes is a filter, allowing it to distinguish bioluminescent light from the faint residual downwelling sunlight, helping it identify prey by the type of light they produce. The gulper eel, Eurypharynx pelecanoides, is a different kind of visual surprise. It appears to be mostly mouth. The body tapers to a very slender tail, but the head and lower jaw are enormous, a vast elastic structure that can open to an extraordinary angle, the lower jaw dropping open like a pelican's pouch, creating a bag-like space capable of engulfing prey considerably larger than the eel itself. It can also inflate itself with water when threatened, making itself appear much larger as a defensive display. The snipe eel, Nemicthis scolopaceus, has a different unusual jaw arrangement. Its jaws are extremely long, longer than the rest of its body, and they curve gently away from each other so that they cannot be closed along most of their length. The function appears to be trapping. The long jaws are studded with tiny hooked teeth, and when a shrimp's long antennae make contact, they become entangled in the teeth, and the shrimp cannot pull free. The eel does not bite or grab so much as it snares. The tripod fish, Bathypteroa grallator, does something that no surface fish does. It stands. The tripod fish has 3 elongated fin rays, 2 extending from the pelvic fins and 1 from the lower caudal fin, that it uses to prop itself above the sediment of the sea floor, raising its body into the water column. It orients into the current and remains perfectly still, using sensory organs in its fins and along its body to detect the water movements produced by approaching prey. It is, in the most literal sense, a fish that has replaced active hunting with the patience of a waiting sensor system. The tripod fish is also a simultaneous hermaphrodite. Each individual produces both eggs and sperm simultaneously. In an environment as sparse as the deep sea, where the chances of encountering another individual of the same species are low, this arrangement ensures that any encounter between 2 tripod fish is potentially a reproductive encounter. Neither individual needs to be a specific sex. Both can reproduce. The goblin shark, Mitsucarina austoni, carries the deep sea's tendency toward extreme predatory structures to a different conclusion. Its snout is elongated and flattened into a distinctive blade-like shape, studded with sensory organs capable of detecting the electrical fields produced by other living organisms. When prey is detected and the goblin shark moves to capture it, its jaw can project forward in a slingshot-like motion powered by elastic connective tissue, extending up to 3 centimeters beyond the tip of the snout. The motion is extremely fast, too fast to follow at normal video playback speed, requiring slow-motion footage to observe properly. One reason may be that the goblin shark's long snout, while excellent for sensing prey, also keeps the mouth some distance from the detected target. The projecting jaw compensates for this by throwing the mouth forward to close the gap. The goblin shark's lineage, the family Mitsucarinidae, has a fossil record extending back approximately 125 million years. The living species is morphologically very similar to ancient fossil forms, making it a textbook example of what biologists call a living fossil, a lineage whose basic form has been conserved across enormous spans of geological time. The oarfish, Regalicus glesner, is the world's longest bony fish, reaching documented lengths of 8 meters and possibly longer. It is silver, ribbon-like, with a distinctive red crest running along its back. It lives in the mesopelagic zone and is rarely seen. But when sick or dying, it sometimes comes to the surface or washes up on beaches. And its long silver form has been, across many centuries and many cultures, the source of sea serpent accounts. The bristlemouth fish, family Gonostomatidae, may be the most abundant vertebrate on Earth, with populations estimated in the hundreds of billions in the world's mesopelagic zone. They are a few centimeters long, dark-colored, with rows of photophores along the underside. They are rarely seen because they live at depth and are individually tiny, but collectively they represent an enormous component of the mesopelagic ecosystem. The deep sea has produced these shapes and hundreds more like them in response to the specific, consistent pressures of its environment. It has done so over hundreds of millions of years, refining each solution with the patient precision of evolution working in a stable environment. And it has done so with light, producing light in forms and for purposes we have only begun to catalogue. That variety of light and its many creators is where we are going next. The flashlight fish, Anomalops catoptron, is named for the most immediately obvious feature of its biology. Beneath each eye, it has a kidney-shaped organ containing bioluminescent bacteria, the same basic principle as the anglerfish's lure, but deployed very differently. The flashlight fish can switch the light off by rotating the organ so that its black backing faces outward, hiding the glow. It can then rotate it back to flash on. In the darkness of the deep sea, the fish uses this on-off flashing to communicate with other flashlight fish. The specific patterns of flashing potentially carrying information about identity, location, or readiness to mate, and possibly to attract the small invertebrates on which it feeds. The cookie-cutter shark, Aecistius brasiliensis, uses bioluminescence in a manner unique among sharks and, as far as is known, unique among predators generally. The cookie cutter is a small shark, less than 60 centimeters, that spends the daylight hours at mesopelagic depths and rises towards shallower water at night. Its entire underside is covered in bioluminescent photophores, except for a dark band of non-bioluminescent skin around its neck, like a collar. When viewed from below, the bioluminescent underside creates a glowing outline, but the dark collar breaks that outline, creating the visual impression of a small fish silhouette against the background light. Researchers believe that larger predators, tuna, dolphins, large sharks, even whales, may be attracted to this apparent small fish silhouette from below. As they approach to investigate or prey upon what appears to be a small fish, the cookie-cutter shark bites them. It uses its circular cutting teeth and rotates its body to remove a perfect plug of flesh, a cookie-shaped bite, typically 5 to 10 centimeters in diameter, and swims away. Cookie-cutter shark bite scars have been found on dolphins, whales, tuna, billfish, seals, and other large marine animals. They have been found on the rubber sonar domes of military submarines. The bites leave a distinctive circular scar, the signature of a small shark that uses its own bioluminescent body as a lure to attract prey much larger than itself. The Atolla jellyfish, Atolla weevillei, has already been mentioned in the context of the burglar alarm strategy, but it deserves a fuller description. The atolla is a disc-shaped, deep red jellyfish of the bathypelagic zone, typically 10 to 15 centimeters in diameter with a long trailing tentacle. When it is grabbed or severely disturbed, it produces a spinning circular display of bioluminescent blue light, a wheel of light rotating around the bell of the jellyfish, pulsing and expanding. The display is dramatic enough to attract other predators to investigate, and the predator that grabbed the atoller may find itself threatened by a larger arrival, releasing the atoller in the process. The deep-sea worm Swima bombiviridis approaches the problem of escape differently. Along its body are small sacs, spherical, attached by short stalks, that glow green when released. When the worm is disturbed or threatened, it can detach several of these sacs simultaneously, releasing a handful of glowing green decoys that drift through the water while the worm retreats into the darkness. The decoys glow for several seconds, long enough for the worm to escape. The detached segments are not permanently lost. They regenerate. The firefly squid, Wattersinia scintillans, creates perhaps the most visually spectacular bioluminescent displays of any deep-sea organism that humans have observed directly. Across its mantle, arms and tentacles, it has thousands of photophores that can be activated in complex patterns. For most of the year, it lives at mesopelagic depths in the Pacific, using these displays for communication and possibly mate attraction. But each spring, in Toyama Bay on the western coast of Japan, vast numbers of firefly squid migrate to shallow water to spawn, and their combined bioluminescent displays illuminate the surface of the bay in blue light visible to observers on boats above. Researchers believe that bioluminescence has evolved independently in the deep sea, not once or twice, but many dozens of times, in different groups of fish, in different lineages of squid and octopus, in jellyfish, in bacteria, in single-celled organisms, in worms. Each independent evolution used slightly different chemistry, different light-producing structures, different mechanisms of control. But the outcome was the same. Light produced by a living thing, used to navigate, hunt, communicate, or defend in an environment where darkness is the fundamental condition. Scientists have also observed something intriguing. In some locations and on some occasions, multiple deep-sea organisms appear to synchronize their bioluminescent displays. How this synchronization occurs and what purpose it serves is not fully understood. It may be coincidental, multiple organisms responding to the same stimulus simultaneously. It may be genuinely coordinated, organisms responding to each other's displays in a chain reaction. The deep sea keeps some of its mysteries close. What all of this bioluminescent diversity reveals is that the deep sea is not simply a dark environment in which organisms struggle to cope. It is an environment that has been actively reshaped by the organisms that live in it, transformed by millions of years of bioluminescent evolution into a world with its own light ecology, its own visual communication system, its own rules for what light means and what it does. And within this world, among the most sophisticated users of the environment's possibilities are the creatures we turn to next, the cephalopods of the deep, whose behavioral complexity sets them apart from almost every other invertebrate group. That is where we are going next. The anglerfish's bioluminescent lure is famous, but the anglerfish is far from alone in using light as a tool in the deep ocean. Across dozens of different lineages, fish and squid and jellyfish and worms and shrimp, bioluminescence has been put to use in an extraordinary variety of ways. Each one is a different answer to the question of how to survive and find resources in a world of permanent darkness. In the deep ocean, where most biological processes are slow growth, slow metabolism, slow movement, the cephalopods are something of an exception. Squid and octopuses are among the most behaviorally dynamic organisms in the deep sea. They change color, they change shape, they change texture, they produce bioluminescent displays, they hunt with speed and intelligence, and they belong to a group, the class Cephalopoda, that has been exploring the possibilities of the invertebrate body plan with remarkable creativity for more than 500 million years. None of the deep-sea cephalopods is more misnamed than the vampire squid, Vampiroteuthis infernalis, which translates, with considerable drama, as vampire squid from hell, is in fact a gentle filter feeder of the deep ocean's most productive current, and it eats marine snow. The vampire squid was first described in 1903 and was initially classified as an octopus, its 8 arms and 2 long retractile filaments being interpreted as octopus-like features. Later examination revealed characteristics of both squid and octopus, but belonging to neither group. The vampire squid occupies its own distinct order, Vampiromorphida, a lineage that separated from both squid and octopus ancestors many millions of years ago and has since followed its own evolutionary path. The name comes from the webbing between its arms, which resembles a dark cloak when spread, and from the red coloration of its body and the blue bioluminescent dots that stud its surface. It is undeniably dramatic in appearance. In character, it is notably peaceful. The vampire squid's primary food is marine snow, the organic particles drifting down from above. It collects these particles using 2 long sticky filaments that it trails through the water, gathering organic material and drawing it toward its mouth. It is, as far as is known, the only cephalopod that does not hunt living prey. In the oxygen minimum zone, where it often lives, a layer of very low oxygen water in the mesopelagic zone, it drifts slowly, collecting its snow, with a metabolism reduced to the minimum needed to sustain it. When threatened, the vampire squid has 2 responses available. It can spread its webbed arms outward and curl them back over its body, turning itself inside out, essentially, exposing either the dark outer surface of its mantle, making it nearly invisible in the darkness, or the inner surface of its arms, which are covered in flexible spines that may deter predators. It can also produce a bioluminescent display from the tips of its arms, blue pulses of light that may confuse or distract predators. The Dumbo octopus, various species of the genus Grimpoteuthis, represents the deepest living known octopus, having been observed at depths of up to 7,000 meters. It is named for the 2 large, ear-like fins that extend from the sides of its mantle, extensions of the mantle tissue that the octopus flaps to propel itself through the water, like an animal flying slowly through the ocean in slow motion. It is not a fast creature. It drifts and flaps with a quality of patient unhurriedness, that seems entirely appropriate for the deep sea environment it inhabits. The Dumbo octopus can flush color changes across its mantle in rapid waves and can extend or retract its arms to change its overall shape. But it appears to use these abilities more for communication and orientation than for active camouflage hunting, in the way that shallower octopus species do. The telescope octopus, Amphitrite pelagicus, is almost entirely transparent. Its mantle, its arms, its internal organs, all are visible through colorless tissue. Only its eyes provide visual contrast, and those eyes are tubular, projecting forward from the mantle like two small telescopes. The telescope octopus is one of the most transparent large animals known. It achieves invisibility not by matching its background, but by becoming part of it, allowing light to pass through its body as if it were not there. Some deep-sea squid have taken the challenge of the deep's two different visual tasks, detecting silhouettes from above and watching the darkness below, and solved it with an unusual anatomical arrangement. The so-called cockeyed squid, including species of the genus Histiotuthis, have eyes of two different sizes. One eye is large, oriented upward, adapted to detecting the faint silhouettes of prey or predators against the residual downwelling light from above. The other eye is much smaller, oriented downward, perhaps adapted for detecting bioluminescent flashes in the darkness below. One organism, two eyes, two different optical systems, two different visual tasks solved simultaneously by a single bilateral creature willing to sacrifice the symmetry that most animals maintain. Cephalopod skin is one of the most complex and versatile surfaces in the animal kingdom. It contains chromatophores, pigment-filled cells that can expand or contract under muscular control to change skin color within fractions of a second. It contains iridophores, cells that produce structural coloration by manipulating light at a nanoscale. And it contains papillae, muscular projections that can be raised to create a rough, textured surface or lowered to produce a smooth one. The deep-sea Humboldt squid has been observed producing complex, patterned bioluminescent flashes across its skin surface during group interactions, displays that researchers believe carry information, but whose content remains undeciphered. The cephalopods of the deep ocean are, in sum, the most behaviorally sophisticated of the deep sea's invertebrate inhabitants. Organisms with complex sensory systems, flexible behavior, and skin surfaces capable of rapid and complex communication. They are as different from the slow, minimal, energy-conservative model of many deep-sea organisms as it is possible to be, while still living in the same environment. And they share that environment with invertebrates of a very different character, organisms that have found other, equally remarkable ways to survive in the deep ocean's unique conditions. Those organisms are where we are going next. The deep ocean's invertebrates, the animals that are not fish and not cephalopods, represent some of the most biologically distinctive organisms on Earth. They include some of the largest animals ever described, some of the most structurally unusual, and some whose biology is so unlike anything in the surface world that they require entirely new frameworks to understand. Let us begin with the largest, the siphonophore, a group of colonial organisms in the class Hydrozoa. related to jellyfish and hydroids, is known in the deep sea to reach extraordinary lengths. Praia dubia, a siphonophore found in the mesopelagic and upper bathypelagic zones, has been measured at 40 to 50 meters in length and possibly longer. This would make it potentially the longest animal on Earth, longer than the blue whale, longer than any fish, longer than any squid. But a siphonophore is not, in the conventional sense, one animal. It is a colony, a community of many individual organisms called zooids, each one genetically identical to the others, each one specialized for a specific function. Some zooids are swimming bells, muscular structures that provide propulsion. Some are feeding polyps, structures with tentacles and a mouth and a digestive cavity. Some are defensive structures with nematocysts. Some are reproductive structures producing eggs or sperm. None of these zooids is independently viable. The swimming bells cannot feed. The feeding polyps cannot move. Together they function as a coordinated superorganism, an animal that is also, in a real biological sense, many animals simultaneously. The siphonophore's tentacles, extended by the feeding zooids along its entire length, can reach out through the water to capture prey, which is then shared through a common digestive canal running the length of the colony. A siphonophore 40 meters long, fishing the water with tentacles extending from its entire length, is a formidable predator in the mesopelagic zone, despite being individually composed of organisms too small to see without magnification. Very different in scale, but no less remarkable, are the zombie worms, Oceanography species, discovered in 2004 on whale falls in Monterey Bay. Zombie worms are polychaete worms, members of the same group as earthworms and ragworms, that have evolved a remarkable and entirely unique way of life. They bore into whale bones using root-like structures that penetrate the bone matrix. They extract the energy-rich fats and proteins stored within whale bones. And they do all of this without a mouth, without a stomach, without any digestive organ of any kind. Instead, within their roots, they harbor symbiotic bacteria that do the digesting for them. Bacteria that break down the bone fats and proteins and provide the products to the worm. The worm, in turn, provides the bacteria with oxygen and a substrate. The relationship is so intimate that neither partner could survive without the other. The visible portion of the zombie worm, the trunk and the feathery plume that extends into the water above the bone, is female. The males are microscopic, barely visible without a microscope, and live inside the female's tube, sometimes dozens of them per female, providing sperm when needed. The entire reproductive strategy of the zombie worm reflects the deep sea's consistent tendency to use every available resource with maximum efficiency. Sea cucumbers, class Holothuriidea, have evolved a defense mechanism of remarkable commitment. When threatened by a predator, some sea cucumber species can expel their internal organs, respiratory trees, gonads, digestive systems, through their body wall. The expelled organs may entangle or confuse the predator. The sea cucumber then regenerates the lost organs over the following weeks. It is a strategy that would seem extraordinary in any context. In the deep sea, where food is scarce and encounters are rare, it represents a calculated trade. Sacrifice the organs, survive the encounter, regrow in peace. The pearlfish, carapace species, has found a living arrangement of unusual intimacy. It lives inside the body cavity of sea cucumbers, entering and exiting through the anus. During the day, it resides within the sea cucumber's internal space. At night, it emerges to feed on small invertebrates. The sea cucumber appears to tolerate this arrangement without apparent harm. The pearlfish has found, in the body of another organism, the shelter that the deep sea's sparse substrate makes difficult to find. The deep sea jellyfish, Deepstaria enigmatica, is one of the more visually striking invertebrates in the deep ocean. Rather than the bell and tentacle form of most familiar jellyfish, Deepstaria is an almost structureless bag, a thin, translucent, irregularly shaped sheet of tissue that can spread to nearly a meter across and that flows through the water with the quality of a slowly drifting fabric. It captures prey by enveloping it, the sheet closing around whatever it contacts, trapping the prey within its volume. Sea angels, pteropod mollusks in the genus Cleone, are tiny, transparent, and predatory in a way that seems almost theatrical. Their prey is the sea butterfly, another pteropod mollusk, and they hunt it using specialized tentacles called buccal cones that are normally retracted inside the sea angel's body, invisible. When the sea angel encounters a sea butterfly, the buccal cones shoot outward from inside its head with remarkable speed, seizing the prey before it can react. The deep sea's invertebrates include organisms that have remained essentially unchanged for 500 million years. The preapellid worms, whose fossil record from the Cambrian period shows essentially the same body plan as living species, and organisms discovered only in the last 2 decades, like the zombie worm, whose biology required an entirely new understanding of how animals can make a living. These are not simple organisms. They are the products of evolution working in a particular environment over vast spans of time, producing solutions that the surface world, for all its complexity, has not needed to find. Among the oldest solutions in the deep ocean belong to lineages so ancient that they connect the deep sea to the very earliest chapters of vertebrate evolution. Those ancient hunters are where we are going next. The deep ocean, with its stable conditions, constant cold, constant darkness, constant pressure, an environment that has changed very little over geological time, has served as a refuge for lineages that have been replaced or displaced in the shallower, warmer, more variable environments above. In the deep sea, certain ancient lineages have persisted. They have not needed to change dramatically because their environment has not changed dramatically. They are living connections to deep evolutionary time. Organisms whose basic forms were established tens or hundreds of millions of years ago, and that continue in the deep ocean's patient darkness to live very much as their ancestors did. None illustrates this more compellingly than the Greenland shark. The Greenland shark, Somniosus microcephalus, is a large, slow-moving shark of the cold, deep waters of the North Atlantic and Arctic Ocean. It is not a particularly striking animal in appearance. Brownish-grey, thick-bodied, with small eyes that are often partially obscured by a parasitic copepod that attaches to the cornea. It moves slowly, with a leisurely undulation of its tail, and it appears in no hurry about anything. In 2016, a research team published the results of radiocarbon dating of proteins in the lenses of Greenland shark eyes. The lens of the eye is composed of proteins that form during embryonic development and are never subsequently replaced or metabolized. They are laid down once at the beginning of life and remain unchanged. By measuring the radiocarbon content of these proteins, the ratio of carbon-14 to carbon-12, which changes predictably over time as radiocarbon decays, the researchers were able to estimate the age of individual sharks. What they found was remarkable. The oldest shark they dated was estimated to be approximately 392 years old, with an uncertainty range that extended above 400 years. Other large individuals in the study were estimated to be well over 200 years old. The Greenland shark appears to be the longest-lived vertebrate animal on Earth, older than the bowhead whale, older than the ocean quahog clam, older by a considerable margin than any other fish. A Greenland shark alive today may have been born in the early 17th century. It was a juvenile when Isaac Newton was formulating the laws of motion. It has been drifting through cold, deep water for longer than most countries currently existing have been independent nations. Its slow, cold life, its slow metabolism and slow growth rate of approximately 1 centimeter per year has extended its existence across an expanse of time that makes human civilization seem brief. Interestingly, the Greenland shark may not reach sexual maturity until it is approximately 150 years old. An organism that does not reproduce until well past a century of age is one whose individual existence spans the entire recorded history of most human institutions. This extraordinary delay is a consequence of its extraordinarily slow metabolism in its cold, deep environment. The same conditions that allow it to live so long also delay every stage of its development. The goblin shark, Mitsukurina austoni, is another deep-sea creature whose lineage extends far into geological time. It belongs to the family Mitsukurinidae, whose fossil record extends back approximately 125 million years, to the Cretaceous period, when dinosaurs still lived on land. The living goblin shark is morphologically very similar to these ancient fossil relatives, making it one of the most compelling examples of evolutionary conservatism known among fish. The goblin shark is pale pink in color, its skin so thin that the blood vessels beneath give it a faint rosy hue, with a long, flattened snout and the remarkable projecting jaw mechanism described in the section on predatory fish. The name in Japanese, Tenguzame, or Tengu shark, refers to the Tengu of Japanese mythology, spirits with long noses. The snout is distinctive enough to have warranted mythological association. The Chimaera, also called the ratfish or ghost shark, represents another ancient lineage. The class Holocephali, of which chimaeras are the living representatives, diverged from the lineage leading to modern sharks and rays approximately 400 million years ago, before the first land vertebrates had evolved, before the Permian extinction, before the dinosaurs. 400 million years is a span of geological time so vast that the arrangement of the continents at its beginning would be unrecognizable today. Chimaeras have been producing chimaeras for all of that time. The living chimaera has a distinctive appearance. large green eyes, a rabbit-like snout, a venomous spine in front of the first dorsal fin, and a body that tapers to a long, rat-like tail. It is found in deep, cold waters worldwide, typically at depths of 200 to 2,000 meters. It feeds on hard-shelled invertebrates, crushing shells with plate-like dental structures rather than the individual teeth of true sharks. The cookie-cutter shark, encountered in the bioluminescence section for its remarkable use of bioluminescent body patterns, is a different kind of ancient predator. Less than 60 centimeters long, it preys on animals many times its own size, using a remarkably specialized feeding mechanism. Circular cutting teeth that remove a perfect plug of flesh when the shark bites and rotates. Cookie cutter bite scars have been documented on a remarkable range of large marine animals, from dolphins and great whales to tuna to seals, and even on the rubber sonar domes of military submarines, which the shark apparently bit in the mistaken belief that they were large marine animals. The deep-sea orange roughy, commercially fished and sold in markets worldwide, can live for 150 years. Researchers believe this extreme longevity reflects the same deep-sea pattern seen in the Greenland shark. Cold temperatures and slow metabolism extending lifespan far beyond what surface relatives achieve. The fishery for orange roughy became commercially significant in the 1970s and 1980s, before the fish's longevity was well understood. Because the fish reproduces slowly and matures late, populations depleted by fishing recover very slowly, a consequence of ancient life history patterns encountered by modern industrial fisheries. The deep sea, then, is not only a habitat for strange and specialized creatures. It is a living archive of evolutionary history, a place where ancient lineages have found refuge in stable conditions, persisting while the world above changed enormously around them. And in some parts of the deep sea, the most ancient relationship of all, between life and energy, has been reimagined from its foundations. That reimagining is where we are going next. On the 18th of February, 1977, the research submersible Alvin descended to the seafloor near the Galapagos Rift in the eastern Pacific Ocean, approximately 2,500 meters below the surface. The scientists aboard were geologists. They were looking for evidence of hydrothermal activity, places where the heat of the Earth's interior was warming seawater, percolating through cracks in the ocean floor. They were expecting to find warm water and perhaps some chemical anomalies. What they found instead was a community of organisms unlike anything previously known to science. And it changed, fundamentally, the understanding of where life can exist and how life can be sustained. The seafloor around the hydrothermal vents was covered in living things. Dense clusters of giant white clams, beds of mussels, mats of white bacteria coating the rock, and rising from the seafloor, in clusters around the warm water seeping from cracks in the rock, were animals that none of the scientists aboard had ever seen. White tubes topped with vivid red plumes, waving gently in the warm water. These were tube worms, and they would prove to be among the most biologically remarkable organisms ever discovered. Riftia pachyptila, the giant tube worm of hydrothermal vents, can reach 3 meters in length. It has no mouth, it has no stomach, it has no intestine or any digestive organ of any kind. It cannot eat. And yet it grows, one of the fastest growing marine invertebrates known, capable of adding up to 85 centimeters to its length in a single year under favorable conditions. The mechanism by which a 3-meter animal with no digestive system sustains itself is one of the more elegant biological arrangements known. The red plume that extends from the top of the tube, vivid red because it contains hemoglobin, is the tube worm's functional interface with the chemical environment of the vent. It absorbs hydrogen sulfide and oxygen from the vent water, binding them to the hemoglobin and transporting them through the bloodstream to a specialized organ that occupies most of the tube worm's body cavity. That organ is called the trophosome, and it is packed with chemosynthetic bacteria. These bacteria use the hydrogen sulfide as an energy source, oxidizing it to produce organic compounds in exactly the same way that photosynthetic organisms use sunlight. The organic compounds produced by the bacteria nourish the tube worm. The tube worm provides the bacteria with hydrogen sulfide, oxygen, and carbon dioxide. Neither can survive without the other. This process, chemosynthesis, had been described theoretically before the discovery of hydrothermal vents, but the vents provided its first vivid biological demonstration. Here was an entire food web sustained not by the sun, but by the chemical energy of the Earth's interior. Here was a thriving community of organisms in a place with no light, no photosynthesis, no connection of any kind to the surface world above. And yet the biomass per unit area was comparable to that of highly productive surface ecosystems. The community around a hydrothermal vent is not sparse. It is dense. Tube worms stand in clusters. Giant clams, Calyptogena magnifica, open their white shells in the warm water, their tissue brick red with the hemoglobin they use to transport hydrogen sulfide to their own internal chemosynthetic bacteria. Pale crabs pick at surfaces and at each other. Pale fish dart through the clusters of tube worms. It is a full ecosystem. Prey and predators and detritus consumers and bacteria, sustained by a crack in the seafloor. The yeti crab, Kiwa hirsuta, discovered near hydrothermal vents in the Pacific, has evolved its own approach to vent-based nutrition. Its claws are covered in long, silky hairs, setae, that harbor chemosynthetic bacteria. The crab waves its claws in the warm hydrogen sulfide-rich water emerging from the vent, exposing the bacteria to the chemical energy source they need to grow. It then grazes on the bacteria it has cultivated, a farming strategy of unusual intimacy between an animal and its microbial crop. Cold seeps, discovered in the Gulf of Mexico in 1984, provide a different but related chemosynthetic ecosystem, where hydrothermal vents are heated by magma and produce superheated water. Cold seeps release methane and hydrogen sulfide at ambient temperature, slowly seeping from geological deposits below the sea floor. The same chemosynthetic principles apply. Bacteria using the chemical energy of methane and sulfide to produce organic compounds, forming the base of a food web of mussels, clams, tube worms, and associated organisms. Whale falls complete a third kind of chemosynthetic ecosystem. When a large whale dies and its body sinks to the sea floor, it becomes a resource of enormous temporary richness. The carcass passes through several ecological stages over decades. In the final stage, the sulfophilic stage. The fats within the whale's bones are decomposed by anaerobic bacteria, releasing hydrogen sulfide into the surrounding sediment. Chemosynthetic bacteria colonize the bones, and a community similar to that of a cold seep develops around the whale skeleton, sustained by the sulfide produced by the decomposing whale, long after the whale's soft tissues have been consumed. The zombie worms, Ocedax, are inhabitants of this sulfophilic stage, boring into the bones and accessing the fats within. They are, in a sense, the interface between the whale fall and the chemosynthetic community that develops from it. Scientists have found that the organisms of hydrothermal vent and cold seep communities are of considerable interest in the context of the search for life on other worlds. Several moons of the outer solar system, Jupiter's Europa and Saturn's Enceladus, are thought to have liquid water oceans beneath their icy surfaces, possibly with hydrothermal activity on their sea floors. The discovery that life can thrive in permanently dark, high-pressure, chemosynthetically sustained environments on Earth has made these ocean worlds more interesting as potential habitats for life. The Lost City vent field in the Atlantic, featuring white carbonate towers up to 60 meters tall, produces alkaline rather than acidic fluids and supports a distinctive microbial community. Some researchers have proposed that systems similar to the Lost City may represent the conditions under which life on Earth first arose. The alkaline chemistry and porous mineral structures of the vents providing a template for the first biochemical reactions. The deep sea, then, is not merely a habitat for life. It is a place where the most fundamental question about life, how it produces and uses energy, has been answered differently from the surface world, and where those different answers have produced ecosystems of extraordinary richness in conditions that, by every surface world standard, should be completely hostile to living things. What these ecosystems reveal, and what the entirety of tonight's journey reveals, is something worth carrying into the quiet of the night. That is where we are going next, and where we will rest. There is nothing left to explain. The zones of the deep ocean have been described. The twilight mesopelagic, the permanent midnight of the bathypelagic, the abyssal depths, and the hadal extremes. The history of discovery has been traced from the Azoic hypothesis to the Challenger expedition, from Beebe's porthole to the ROV, from the mythology of the Kraken to the 2004 photograph of a living giant squid on a camera line 900 meters below the Pacific. The bioluminescence has been described, the 76% of deep-sea organisms producing their own light, the blue and green that travel furthest, the private red of the dragonfish, the burglar alarm of the atoller jellyfish, the green bombs of the deep-sea worm released into the darkness as decoys, the bodies built for the deep, gelatinous and pressure-adapted and TMAO-filled and oil-buoyed, have been described. The food adaptations, the slow metabolism, the enormous mouth, the expandable stomach, the years-long fast of the giant isopod have been described. The anglerfish has been visited. The glowing esker in the permanent darkness, the tiny male following chemical gradients across the vast dark ocean to find his female, the permanent fusion of tissues that converts two independent organisms into one interdependent unit. The giant squid has been visited. Its dinner plate eye evolved to detect the bioluminescent approach of sperm whales. Its existence confirmed as a living animal only 2 decades ago, despite centuries of mythology. The predatory fish have been visited: viperfish and dragonfish, and fangtooth and stoplight loosejaw. Each one a different answer to the question of how to hunt in complete darkness with rare and unpredictable food. The strange-shaped fish have been described: the barrel eye with its rotating eyes in a transparent dome, the goblin shark firing its jaw forward in a motion too fast for normal video, the tripod fish standing patiently on its fin rays, waiting for the vibration of approaching prey. The other light makers have been visited: the flashlight fish with its rotatable bacterial organs, the cookie-cutter shark using its own glowing body as a lure, the firefly squid illuminating a Japanese bay each spring. The cephalopods have been visited: the vampire squid drifting gently through the oxygen minimum zone collecting marine snow, the telescope octopus nearly transparent, the cockeyed squid with its 2 different eyes for 2 different visual tasks. The worms and jellies and colonial superorganisms have been described: the siphonophore, 50 meters long, composed of thousands of individual organisms, each specialized for one function: the zombie worm dissolving whale bones with bacteria in the absence of any digestive system, the sea angel shooting its buccal cones from inside its own head to seize a sea butterfly. The ancient lineages have been visited: the Greenland shark drifting through Arctic water at the age of 3 or 4 centuries, the goblin shark, whose family line extends to the age of dinosaurs, the Chimaera, whose ancestors diverged from sharks before the first vertebrate had set foot on land. And the chemosynthetic ecosystems have been described: the hydrothermal vent community, sustained entirely by the chemical energy of the Earth's interior, the tube worm with no mouth and no stomach sustained by bacteria within its body, the yeti crab waving its furry claws in the warm vent water to cultivate its microbial crop. All of that is there now, somewhere in the mind, held gently, without urgency. What remains is simply to rest in it. Right now, as you rest, the deep Pacific Ocean is carrying on its patient business in the permanent dark. In the midnight zone, 1,000 meters below the surface, in complete and absolute darkness, an anglerfish drifts. The esker above her mouth glows blue, a single point of living light in all that darkness. It has been glowing, generation after generation, in the deep ocean for tens of millions of years. In the mesopelagic zone, a vampire squid drifts on its fins through the oxygen minimum zone, trailing its sticky filaments through the water, collecting the slow rain of marine snow that falls from the world above. It does this gently, patiently, making use of what is available. There is nothing hurried about it. In the deep, cold water of the Arctic, a Greenland shark moves slowly through the darkness, perhaps 300 years old, perhaps 400. It does not know how long it has been in this water, in this cold and this dark. It knows only the slow chemical signals that guide it toward food, and the patient movement through a world that has been its world for longer than any nation on the surface above has existed. In the midnight zone, a giant squid turns its enormous eye, 30 centimeters across, the largest eye of any living animal, toward a disturbance in the water, a flash of bioluminescence, something large approaching. The eye takes in every photon available. The squid assesses. The world of the deep sea is not empty. It is full of information, of communication, of predator and prey relationships of extraordinary sophistication. All of it carried out in the language of bioluminescent light, of chemical gradients, of pressure waves in the dark water. Researchers believe that most of the deep ocean's creatures will never be seen by any human being. The ROV footage and the submersible observations represent a tiny sample, a few dozen sites revisited over decades in an ocean that covers most of the planet. The majority of deep-sea organisms will live and die in the darkness they were born to, without any human awareness of their existence. This is not a loss, exactly. It is simply the nature of a world as large and as old as the deep ocean. It has been doing what it does, producing bioluminescent displays, sustaining chemosynthetic communities, growing ancient sharks and patient tube worms, without any need for human observation. The bioluminescent displays are happening now, right now, in the permanent darkness of the deep Atlantic and the deep Pacific and the deep Southern Ocean. Flashes and glows and pulses of living light, produced by organisms that are going about the slow, careful, extraordinarily adapted business of deep sea life, as they have been doing for hundreds of millions of years. The deep ocean is patient. Its creatures are patient. The anglerfish waits. The tripod fish waits. The Greenland shark waits. There is something restful in that image. A world of patient waiting, of slow movement, of long lives lived with quiet efficiency in the permanent dark. Sleep well.